All right. Hello. So, for this lecture, we are gonna talk about DNA, specifically the structure of DNA. So the exact molecular atomic structure, the bonds, the chemical groups that are involved, um, as well as an in-depth look at the mechanism of DNA replication. So DNA replication, being the process that cells undergo, mostly, if not always, prior to cell division. So before the cell can divide and produce an identical, genetically identical uh, uh, copy, clone really, of that cell. Importantly, DNA replication actually also occurs before meiosis. So producing an identical cell to the original parent cell, that would be a mitotic process. That would be mitosis, right? But DNA replication will also occur prior to meiosis, which, of course, actually will generate genetically unique cells. But the point is this process of DNA replication is going to occur before any kind of cell division, so that each daughter cell is going to receive the appropriate amount and the appropriate type of DNA molecules, right? So let's dive in, and we're going to start with the structure of DNA and a bit of history behind it as well, and how that structure was ultimately discovered and determined by very prominent scientists in the field of molecular biology. And this was roughly 1950s when this discovery was made. Nobel Prize winning discovery, of course. So, okay, let me get my slides going here. Oops. Okay. Here we go. All right. So our story here begins with these scientists shown on the left side here. And we'll start with the top scientist up here. Her name is Rosalind Franklin. So this is Rosalind, and actually I know I have to angle this now. Here we go. Rosalind Franklin. So prominent, very famous biochemist of the 20th century. And importantly, and again, this was like 1950s, early 1950s, Rosalind Franklin was working in a lab in the UK at King's College in London. So essentially at this time, there was a sort of race to discover among scientists the exact molecular structure of DNA. At this point, they knew that DNA was in fact the genetic material. It was the molecule. It was the type of biological molecule that contained the information that codes for life and is transmitted from generation to generation. But they didn't know what it looked like. They didn't know the bonds, the atoms, the chemical groups involved. So the very first image of DNA is actually shown here as well. Actually, I can use my mouse. Here it is. One of the most famous images in biology. And it's kind of like, you can see it's kind of this X-shaped image. It was produced by a technique called X-ray crystallography. So, okay. So X-ray crystallography, which is actually a technique that's still used today, although it's used much more cautiously, as X-rays are very dangerous, volatile. They, high-level radiation. Ironically, they damage DNA. I just thought of that. Which is why you would wear the lead when you get an X-ray at the dentist. Now, of course, unfortunately, the 1950s, these kinds of things weren't in place. The scientists didn't really understand yet the dangers implicated, even smoking, right? So sadly, tragically, Rosalind Franklin passed away from cancer a few years, I believe, after generating this data and writing her paper. And we'll get to that a little bit because it's a bit of a controversial story and it's a tragic one at that. But anyways, she produced this image, right? And it's this X shape, right? Well, DNA doesn't really look like an X. It's actually a top-down view of the DNA molecule, which, as you might know, it's a very well-known image and structure. I'm sure you've seen it all of your lives, but it's this double helix twisted kind of ladder structure. So if you do that, kind of looks like this, right? With the rungs of the ladder connecting in between. And so it was a top-down view, which would look like an X. Okay. Okay, so that brings me to the next part of the story, which is the bottom two scientists. Now, these guys are named James Watson and Francis Crick. So we've got James Watson and Francis Crick. Now, they were also trying to discover the structure of DNA. And they actually worked in a neighboring lab at King's College, a neighboring lab to Rosalind Franklin's lab, okay? And here's where the story gets kind of controversial in many ways. So Watson was creeping around, walking around the hallways, and he noticed this image. He saw the X-ray crystallography image from Franklin's group. And he also found an unpublished report from them as well that was summarizing Franklin's work. And essentially, Rosalind Franklin and her group concluded that, as we'll talk about, this backbone, so kind of the outer strands of DNA, they were composed of sugars and phosphates, as we'll see. And that was contrary to their working model. So they kind of piggybacked, is my point. They piggybacked off of Franklin's work. And from that, they were able to elucidate the exact specific molecular architecture and the exact atomic arrangement of the DNA double helix. And for that, they were awarded the Nobel Prize in 1953. Now, sadly, Rosalind Franklin passed away from cancer before the Nobel Prize was handed out. So that's kind of how they justified not including her in awarding her the prize. So, and that's kind of obviously controversial and messed up in my opinion. So that's kind of the story, right? Now, again, let's describe the DNA molecule as a whole. It is a double-stranded, meaning it's really, well, you could say two chains of molecules that come together, right? And are held together by specific bonds, and we'll take a look at this. But it's a double-stranded nucleic acid. So if you recall the four different categories of molecules that exist in biology and life and organisms, nucleic acids is one of those four categories. It's not a carbohydrate, it's not a lipid, it's not a protein, it's a nucleic acid. And if you recall, and we'll do a bit of review on this because it's been a while, but we're gonna review the monomer unit that makes up nucleic acids. And if you remember, they are called nucleotides. And the overall kind of geometry and shape of the thing is it's a double helix. So a helix being, you ever been to an amusement park? It's kind of a twisty, coily looking thing, right? So it's two strands that are twisted together and form this double helix. Okay. Moving on. Okay, so like I said, let's take a look and review the monomer unit that makes up nucleic acids, such as DNA. And again, it's called a nucleotide, right? As you can see. Oops, I think you can see that. Nucleotide. Okay, so let's review what a nucleotide is made of. And remember the kind of the idea with monomers and polymers is that the giant, large functional polymer form of these molecules like DNA, for example, they're made of hundreds to thousands, and in the case of DNA, really millions and millions of nucleotides connected to one another via covalent bonds. So let's take a look at one single nucleotide and what that looks like. So recall that a nucleotide is composed of three parts. Three parts. So start with parts. The phosphate group. And as you know by now, Professor LaRue always wants you to associate phosphate groups as being negatively charged. So their chemical formula and kind of what makes up a phosphate, you got a P, phosphorus, O, four oxygens. And it always is going to have a negative charge, in some cases, a 2 minus charge. And so let's keep that in mind, that it's our phosphates are always negative. And that plays a lot of cool, interesting roles as far as DNA being this highly negatively charged molecule, because you've got, millions of these phosphate groups that are all negatively charged. And so that actually structure and function being connected again, as I always say, intimately correlated, a really fundamental theme in biology of structure and function. Well, when we do DNA forensics and we run DNA molecules through a gel, we apply an electric current and it actually migrates through the gel primarily because it's negatively charged due to the phosphate group. So there's a lot of interesting applications for that. So our nucleotides are going to have a phosphate. They're going to have a sugar connected to that phosphate. It's a 5-carbon sugar. 5-carbon sugar. And since today we're focusing on DNA, the nucleotides that specifically make up DNA have a specific type of sugar that make up the nucleotides of a DNA molecule. And it's called deoxyribose. And so I'm actually going to color code now. So blue is going to kind of be my indicator that we're talking specifically about DNA. And so for DNA, the sugars that make up nucleotides of DNA, it's called deoxyribose. And this actually is why DNA stands for, as you might know, deoxyribonucleic acid. Okay, well, that makes sense now. Deoxyribo, because of the sugars in the nucleotides that make up DNA, or deoxyribose sugars. And then, of course, it's a nucleic acid, DNA. Last, the third part of a nucleotide is going to be our nitrogenous base. And again, since we're focusing on DNA, we'll talk about the types of nitrogenous bases you can have in a nucleotide of DNA specifically. As we'll see when we look at RNA eventually, a different type of nucleic acid, there's a few subtle slight differences when it comes to the bases and even the 5-carbon sugar. But in DNA, there's 4 different varieties of nitrogenous bases that you can choose from. So there's really, you could say, 4 different nucleotides to choose from based on the base. And so I'm going to, again, color code the nitrogenous base that is unique to DNA. You wouldn't find it in any other nucleic acid like RNA. But there's 4. So you can have an adenine base, but it's, again, not unique to DNA, so I didn't make it blue. Adenine is found in RNA molecules as well, nucleotides that make up RNA. Now, the next one, thymine, is unique to DNA. And by the way, we can abbreviate these. Adenine can be abbreviated with an A. Thymine, which is only found in DNA. You wouldn't find thymine in RNA, but you can say it's a T. And then the other 2 bases are found in any nucleic acid as well. So those are, writing out of room at a, sorry, this whiteboard isn't super big, but we've got also cytosine. cytosine C, and guanine G. And so actually on the slide here, I have pictured an example of a nucleotide, and it DNA nucleotide, and it has an adenine base, as you can see. But you can see it has a phosphate, the 5-carbon sugar, and the base. Nitrogenous with all the nitrogens, right? Okay. All right, so moving on, you might have noticed that there are some numbers here. Now that'll play a really important role very soon as we look at the structure of the DNA molecule as a whole. But I just want you to keep in mind for now that these numbers are referring to carbon atoms that are part of this sugar, specifically the 5 carbons. 1, 2, 3, 4, 5. And then there's this little dash mark that we refer to as prime. So 1 prime, 2 prime, 3 prime. So we'll talk a bit about that pretty much on the next slide. But just keep that in mind that we're referring to the carbons in the 5-carbon sugar of the nucleotide. Okay. Oh, actually. So let's go ahead before we look at the cool fancy image. We're gonna draw a DNA molecule by ourselves here. So let's do that. All right, so let's look at the structure of DNA. Move this a little. Okay. Structure of DNA that Watson, Crick, and Rosalind Franklin discovered. So DNA structure. Now, of course, like I mentioned, DNA is made of thousands to millions, typically millions of nucleotides. I can't obviously write that many here. So we'll probably do 3 just to show you kind of the general idea. And so again, it's double stranded. So we're gonna start with the strand over here, and then we'll go to the strand there. So nucleotides, right, that are bound together. So we'll do 3 nucleotides. So we'll start, and I'm simplifying here, but we've got a phosphate group that'll be connected to a sugar. Right, so then the sugar, as you saw, is connected to one of the 4 nitrogenous bases. Let's just choose A for this one, adenine. Okay, next nucleotide will have a phosphate again. Sugar. Off the sugar, let's make this a C, cytosine. And then let's do one more nucleotide here, sugar. And let's do a T, thymine here. So that's one strand, right? Okay. As we know, DNA is two strands, two chains of nucleotides that are coming together now. And so let's do the other strand. And importantly, we're not going to start with a phosphate. This will be a very important point, and we'll talk about why. But actually, we're going to just start with the sugar here. And the sugar is going to be connected to a T. Now, why did I choose T? I have no choice. This is called complementary base pairing. Okay, so complementary base pairing. Not complement like you have nice shoes, but complement with an E where they complement one another. And essentially, so complementary base pairing. So in a DNA molecule, whenever you have an A base, it will always be paired to a T and vice versa. You got a T, it'll be with an A with a T. And if you have a C, it's always going to be paired with a G and G with C. So one of my TAs gave me this really fun little way to remember this. Apples are found in trees, cars park in garages. So I thought that was a pretty fun way to remember. But essentially, yeah, so you're going to have an A paired with a T here. Okay. Now, while we're talking about this pairing, what is holding them together? Okay. Well, a familiar friend, it's been a little bit, it's been a while, but if you recall, this very important type of bond that exists between separate molecules that can kind of keep them held together. And we talked a lot about how water molecules do this, but hydrogen bonds are going to connect the two strands of a DNA molecule. And specifically between A's and T's, there's going to be two, and remember we represent them with dashed lines, two hydrogen bonds that form between A's and T's. Sorry, that looks faded. Okay, two hydrogen bonds. Very weak intermolecular forces. Again, important structure function connection because as we will see very soon, actually, DNA is constantly being unwound. The two strands are constantly being separated all the time for replication, for central dogma, which we'll get to another time. But yeah, it's great that there's a weak attraction between the two strands because it kind of like Velcro, we can separate and reconnect very easily. Okay, moving on. Our next nucleotide here, we're going to have a phosphate, of course, sugar, and okay, based on our base pairing, what am I going to put here? We got a C. Where are we going to pair here? That's right, invisible audience. We're going to put a G. Cars parking garages. Now, important difference here is that actually 3 hydrogen bonds form between C's and G's. So 1, 2, 3. So we get 3 hydrogen bonds here. So we got 3 between C's and G's, 2 between A's and T's. Okay. Lastly, we've got another phosphate. Okay, I'm running out of room here, so bear with me. But we'll have a phosphate, a sugar, but oh man, gotta make it a little smaller because very importantly, we'll actually have another phosphate sticking down here because this last nucleotide can't just be one sugar and a base. It needs its phosphate. So I'm just gonna redraw that a little smaller. So let's do phosphate, sugar, phosphate. And of course, off this sugar, I know this isn't really drawn to scale very well. I apologize, but we've got an A that would connect to our T here. And again, it would be two hydrogen bonds that form. All right, moving right along. Really, the last kind of important concept with the structure of DNA and the helix, the double helix, it has to do with those carbons that I was talking about on the sugars. And it has to do with this concept of what we call directionality. Essentially, the two strands of a DNA molecule run in opposite directions. What do I mean by that? I mean that this strand here is going in a certain direction, let's say upward. Well, then this strand would be going down. So it's kind of a silly way of saying that this strand, all the atoms, the bonds, the molecular structure of it is flipped with respect to this strand. If you took this strand and you flipped it upside down, all the atoms, all the bonds, it would mirror, it would be this one exactly. Of course, it has different bases, but the sugar phosphates, all the atoms, you just flip, they are flipped. They are kind of inverse of one another, okay? So how do we represent that? That's what the next slide I really like. I really like it because you can see very clearly what I'm talking about on the right side here. If you see that all of the carbons in the sugars of all the nucleotides are labeled, you can see that the fifth carbon here, you can see my mouse cursor, is towards the top of this strand. Whereas the third carbon of this nucleotide's sugar is towards the bottom. Now, like I said, imagine flipping the thing, and you'll see that you have everything flipped on this side, right? And so you actually have the third carbon sticking up on this side and the fifth sticking down on this side. Now, importantly, the fifth carbon, the 5' carbon of these sugars are connected to the phosphate groups. And that's why there's a phosphate sticking up here, but not down here, and then sticking down here and not up here, if that makes sense. So we actually can refer to the strand that has the phosphate sticking up. This would be your 5' end. And again, this is prime. Your 5' end. Because the fifth carbon is sticking up closest to the end here. Now here, the third carbon is sticking closest to the end. The fifth one is actually here, connected to the phosphate. So the 3' carbon is sticking down. So this is your 3' end here. And in fact, we can also draw in, there's a hydroxyl group, kind of ran out of room, but it's just an OH group. Redraw that really quick. So sticking off of the bound to the 3' carbon is an OH, like that, hydroxyl group. And this is your 3' end. Now, if you look at this side, of course, it's reversed, right? This would be your 3' end sticking up here, and you could draw an OH coming off. And then down here with the phosphate would be your 5' end. So we refer to this phenomenon as DNA is a molecule that we call anti-parallel. The anti-parallel nature of DNA. So anti-parallel meaning the two strands run in opposite directions. They're flipped with respect to one another. Their orientation is reversed. All right. So now that we've looked at the structure of DNA, we can start to get into replication. All right. So let me go ahead and erase this. All right. So DNA replication. Before we dive in, there's this concept of an origin of replication. So if you look along the length of a DNA molecule, there are areas, in some case there'd be just one, which we'll talk about. And they're called origins of replication. They're short stretches of DNA that is just a specific sequence of nucleotides determined by the bases. Like there'd be maybe a 10 or 15 nucleotide stretch, and that's where your replication origin is. That's where replication will occur. So if you take a look at this really cool slide that distinguishes the difference between replication origins in E. coli, for example, versus a eukaryotic cell. So I like this image because you can see, first of all, that bacterial chromosomes, prokaryotic chromosomes, DNA molecules, are circular. You can see it here, right? So you've got this double-stranded DNA, but it connects in a circle, which is kind of interesting. Importantly, with our prokaryotic cells, you'd have one origin of replication from which you would replicate and produce your two identical DNA molecules. On the other hand, in eukaryotes like us and our cells, our DNA molecules are much larger. And so, or our chromosomes, rather, can be upwards of 10 times as big. In the case of a DNA molecule inside eukaryotic cells, they're linear. So you can see that the DNA molecule is actually just a linear shape. And there's actually multiple origins of replication. As you can see here, you've got multiple sites where the DNA is being replicated. You're producing identical DNA strands. Well, that's not technically, I guess what I'm trying to say is you'll end up with two identical daughter DNA molecules, but you're producing, you're starting along the DNA molecule at different places, okay? And eventually they actually fuse together. So of course, what does that mean? It means that even though the DNA molecule is larger in eukaryotes, the process is going to take roughly the same time or shorter because we're starting at multiple places and eventually it fuses together instead of just one spot going out. You're starting at multiple places, so you kind of cut the time shorter. Okay. Now, this image shows as well that there's this concept of how DNA replication works, this mode of replication that we call a semi-conservative mode of replication. So let's define what that means. So semi-conservative DNA replication. Okay, so what does this mean? And we won't get into the nitty-gritty of how this was discovered and the different ideas at the time of how this worked. But essentially, as you can see on the slide, and we'll write this out as well. Here's your, let's look at the E. coli one, for example, on here. So you see this original DNA molecule. It's kind of this dark blue, right? Well, the new strands that are being formed, right, at this origin of replication, they have a light blue color, right? Eventually, when you get your two brand new DNA molecules, you can see that each one has half dark blue, half light blue. Half dark blue, half light blue. So what does that mean? It means that each brand new DNA molecule, they're genetically identical, or they're the exact same, right? But each one contains half of the original parent molecule, and the other half is brand new, newly synthesized, right? And here as well. So we call that being semi-conservative, which again, half of the original DNA molecule is kept or conserved as the template strand, and we'll look at this in detail in a minute, as the template strand for the new complementary strand or DNA strand. So they're kind of like a hybrid of half old, half new. All right. Next up, we've got how this happens, how and the host of enzymes involved, all this good stuff is going to go down. And we're going to draw it out, of course. It's always fun. So as you can see here, you've got what we call these replication bubbles, okay? Which is where the replication is occurring at this origin of replication, right? And you can see that actually that replication proceeds in both directions, right? Here and here. It's replicating both ways. And same as the eukaryote. Each kind of edge here we call a replication fork. So you've got a replication fork here and a replication fork there. That's what we're going to zoom into to see how at the replication fork, how is it synthesizing these new DNA strands that are complementary to that original template parent strand, okay? Oh, actually. Okay, so again, we call this a replication fork. Oop. And this is what we're gonna draw out right now. So let's do that. Okay, so we'll start with a little bit of the parent molecule that's not replicated yet. And then it's gonna kind of do this bell curve looking thing like that. So we'll draw some bases coming off. So 4, 5, 6, 7, 8, 9, 10. We'll do 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And again, this is our replication fork that we're diagramming. Replication. We're going to hop back and forth from the slides to here to kind of define what's happening. And by that, I mean, there's a lot of enzymes that play roles in replicating DNA. And the very first one we're going to talk about is the one that does that, separates the two strands of DNA. This enzyme actually moves along in this way, okay, that way, going that way. It's moving along the DNA and opening it up, separating, breaking those hydrogen bonds and separating the two strands. That enzyme is called helicase. So helicase, I'm going to make it green. Let's make it, let's just put it here. And all these enzymes are kind of like little blobs. So here's our helicase enzyme. And again, helicase is unwinding the double helix. That's its role. Okay. Next up, we have actually something, these aren't enzymes. They're called single-stranded binding proteins, or SSBPs. Now, as we know from hydrogen bonds, opposite partial charges coming together, they want to essentially these strands, they want to come back together, of course, right? So the SSBPs are these little proteins that hook on Just do two, I guess, like that. That hook on to the DNA strands and kind of prevent the strands from coming back together. So that's what they're doing. So these are our single-stranded binding proteins, or SSBPs. And then there's one more kind of funky enzyme that's doing this kind of weird job of, it's called topoisomerase. And essentially, I don't have a rubber band with me. I need to get one. Been meaning to get one to demonstrate this, but essentially there's this phenomenon called supercoiling, or it gets really complex and strange, but essentially, if you have a rubber band and you open it up, right, and start twisting it, like a DNA molecule is twisted up, right? But then, of course, we're trying to open it up. At the ends, it gets a lot tighter, right? So it gets tighter twisting and strain further down along this DNA molecule. And so this is what the textbook says. I don't know how it does this. I guess with a rubber band, you could kind of show that you can break and rejoin and try to relieve that tension. That's what topoisomerase does. So in this class, of course, even in like graduate level, topoisomerase is kind of an oddball of an enzyme, but we'll draw it in as playing its role further down and relieving this strain caused by the opening of the DNA. So let's just kind of put it here. So topoisomerase. Okay, now we got the kind of odd enzymes out of the way. So now we're going to build this thing. And so essentially, enzymes in DNA replication, even in our central dogma that we're going to look at soon, not today, but these enzymes cannot, actually, no, it is just DNA polymerase. I'm sorry, I misspoke. DNA polymerase can ignore the central dogma thing I just said. DNA polymerase cannot initiate DNA replication. It can only extend pre-existing nucleotides and kind of add more nucleotides to that chain. Okay. So what enzyme can initiate things? It's called primase. And so primase, we'll use red, is actually going to get things going here. It's going to start things off. It's going to prime our strand. And it does that by adding this primer. And a primer is actually RNA. It's not DNA, interestingly enough. So primase adds a 5 to 10 nucleotide long RNA molecule, single-stranded. We'll see that in a minute. And it's called a primer. So let's take a look at what that looks like and how primase does this. So let's look at it on let's look at it on this strand first. And so we'll come over here. I know I said 5 to 10 nucleotides long, but let's just kind of represent it here. So we'll have our little RNA primer, right? So this is our primer. And it's added by the enzyme primase. So we can actually draw the enzyme here. Let's do it kind of attached here. So this is our primase adding a primer, and it moved along in this direction and added this primer. Now that I'm looking at the image, in reality, it kind of hooks the entire, it kind of takes up the whole, it kind of clamps on here, but you get the idea. Okay. So again, DNA, to make more DNA, we can't do that, right? These enzymes, they can't just add these nucleotides from scratch. They can only extend from pre-existing nucleotides, right? So the next thing that's going to happen, is DNA polymerase, which is really our main enzyme here. And there's two kinds, which is, we'll get to that. But DNA polymerases are going to extend and add complementary DNA nucleotides to these strands, okay? So let's take a look at what that looks like. So we use blue for DNA. And so essentially, DNA polymerase, let's draw that in first. And let's do that. Actually, let's do it up here. Kind of hook it down, kind of clamps on here. So this is DNA polymerase. Now, specifically, as we'll see, this is going to be called DNA polymerase 3, probably because it was discovered third. Actually, I'm not sure why, but it's called DNA polymerase Roman numeral 3. And what it's going to do is go ahead and add complementary nucleotides to these template DNA nucleotides right after the primer. All the way up here as we keep going. And so it's moving along the strand here, adding these nucleotides, complementary to the template here. And of course, they'd actually eventually start to connect. It's forming a bond between them as well. We do this last one as well. And as you can see, it kind of just glides along and transfers, adds in these complementary nucleotides completely uninterrupted. No problem at all. And for that reason, we actually call this strand the leading strand. We'll talk about what's going on there in just a second. But this is our leading strand, okay? All right. Now we got to talk about this other strand. Okay, actually, before we do that, sorry. Now the other strand's a little more funky. Now this was no problem, okay? Bumper sticker motto phrase, okay? Are you ready? I know you've heard a bunch of them by now from me, but essentially, any polymerase enzyme synthesizes, and this is why we looked at the structure of DNA before this. I'm sorry, reads in the 3 to 5 prime direction, synthesizes 5 to 3. So polymerases, I ran out of room on this whiteboard. Where could I write it? Might have to erase it in a minute, but we'll see. But I'm going to try to squeeze it in here. Polymerases read in the 3 prime to 5 prime direction, comma, synthesize 5 to 3. And I remember that because like when you read a book, you start it, you go in order, right? You don't go from like page 5 to 3. You would go 3, 4, 5. You would go in order, right? So they read 3 to 5. So what do I mean by this in this context? Well, essentially, we can look at the leading strand here first. Let's write in the directionality, the orientation of the DNA molecule here. So if we look at our original, and I think I used green on the other side, so let's do that. This polymerase is going to read the template strand in the 3 to 5 prime direction. So what does that mean? It must mean that starting here is your 3 prime end, and ending here is your 5 prime end. So it's reading 3 to 5, but the new strand that's being synthesized, okay, is anti-parallel. It's complementary and anti-parallel more specifically. And so this would be 5 prime to 3. So it's building 5, synthesizing 5 to 3, but reading 3 to 5. Okay. All right. Maybe some of you deeper thinkers have already know where I'm going with this. But this is the great conundrum and was a Nobel Prize winning discovery by Reiji Okazaki. And essentially, this strand's in trouble. We are moving this way. We are moving along the DNA molecule and replicating it in this way. However, this strand is anti-parallel, and therefore it's going to be 3 prime, right? And we're talking about this template strand. And this would be 5 prime. Now, here's the problem. We are moving, the overall direction of replication is this way. And yet, we've got to replicate this, and we can't go that way. Why not? DNA polymerase cannot read 5 to 3. It can only read and synthesize in this direction, which is the freaking opposite direction of where we're supposed to go, right? So it's kind of like it's got to come this way while we're moving that way. Okay. And so how is this resolved? Kind of in a funky way. What happens is that DNA polymerase 3, so there'll be another one here. Let's draw that. So DNA polymerase 3, actually, of course, I think we'll have to do it. Draw it here. So this is another DNA Pol 3 for short. It's got to move in this direction, away from the replication fork, right? And so, this strand is going to be synthesized discontinuously. The leading strand was just continuous, super easy. Well, this one, we keep moving this way, and so this enzyme's got to keep coming back as we're moving forward, if that makes sense. Because it can only go in this direction, reading 3 to 5, synthesizing 5 to 3. So what happens is primase has to separately prime a bunch of segments, RNA segments. So we'll do one here, for example. So primase is going to come in and prime this, right? Let's do primase. How did I? Oops, I'm sorry. Totally drew that wrong. My bad. So primase would come before DNA Pol because we're going this way. So it would actually do that one. So this is our primase. And it's making this little RNA primer here. And then DNA polymerase can go ahead and work in this direction, synthesizing 5 to 3. Let's add 3 here, okay? But we keep moving forward. That's the problem, right? And he can only go this way. And so now we've got to separately prime. Primase is going to come, another primase is going to prime another short RNA segment so that DNA polymerase 3 can then extend and keep going to finish this. Now, that is why we refer to this as the lagging strand. It's synthesized discontinuously. So this is our lagging strand. Okay. These DNA segments that are like discontinuous in between like this RNA, and it's all really uncoordinated and crazy. Those we call Okazaki fragments after the scientists who discovered this mechanism. So these would be called Okazaki fragments. Sorry, the writing's getting really small because I'm running out of room here. But these are Okazaki fragments. Again, because you got to keep, as we move this way overall, the leading strand is fine. It just keeps going. But this polymerase can only read in this direction, right? So it's going to keep having to come back. Primase, primer, boom, primer, boom. As it tries to keep up and it's lagging behind, right? Okay, I promise we're basically almost done. So there's really just 2 more enzymes that play a key role in this. and I'm sure you can guess what it might be doing, or at least one of them. We can't leave RNA in a DNA molecule. We need to freaking have identical DNA molecules at the end of this, right? And so there's actually another DNA polymerase whose role is to take out the RNA molecule primers and replace them with DNA. And so it should be on here. I guess not. Huh, gotta add it. Okay. But it is on this summary. DNA polymerase 1 is going to take out the RNA nucleotides and replace them with DNA. So we can kind of represent that with like, just like a big X here. Okay, and then another enzyme. Again, it's DNA, but this time it's DNA polymerase 1. So let's make a little blob attached. This is our DNA pol 1. And again, DNA polymerase 1, its function is to take out the RNA primers and replace them with DNA. And it would do it for this one primer up here as well. So you could even do that and kind of connect it. And it'll replace it with DNA nucleotides. Now, that still leaves these gaps in between. So to connect the sugar phosphate backbone, if you will, so like a sugar and a phosphate, to connect them together and catalyze that bond, there's another enzyme, it's our last enzyme we'll talk about, called DNA ligase. And so DNA ligase, so let me show you here. So ligase here is going to connect the backbones. They're going to connect these discontinuous DNA segments, specifically the Okazaki fragments. So ligase is going to come in, and it's going to be DNA. So the enzyme I'll make purple. So let's make it here. This is our DNA ligase. And it's going to connect the discontinuous fragments after DNA polymerase 1, of course, replaces RNA with DNA. Woo, okay, that was a lot. So that's it. That's DNA replication. A lot of enzymes involved. And it's kind of a weird mechanism that evolved here to make sure that, that have to follow this kind of bumper sticker of, we can only go in this direction, reading 3 to 5, synthesizing 5 to 3. And yet again, the overall direction of replication is we're moving down the DNA molecule this way. So it keeps having to come back and synthesize these discontinuous Okazaki fragments. Okay, and then that's it. That's all I got. All right. Hope you enjoyed.